Our protocol provides a practical approach by which mouse spleen and lymph nodes can be used to reflect the dynamic of T-cell response, rather than providing CD8 phenotypic snapshots. Our technique sensitively detects antigen-specific CD8 T cells that are engaged in an ongoing response. Proliferating CD8 T cells are measured without possibly confounding effects of in vivo drug treatment or cell transfer.
Our technique can provide a window on immune dynamics in several settings, for example, in relation to response to vaccination and infections, autoimmune diseases, and cancer immunotherapy.
Prepare fresh 1x permeabilization wash buffer by diluting 10x permeabilization buffer with distilled water and filter it through a 0.45 micrometer filter to eliminate aggregates. Dilute monoclonal antibody Ki-67 FIT-C in 1x permeabilization wash buffer as determined with a titration experiment for a final volume of 100 microliters per sample. Add 3 milliliters of the 1x permeabilization wash buffer to each tube with cells and centrifuge at 400 G for 5 minutes at room temperature. Discard the supernatant. Again, add the 1x permeabilization wash buffer and centrifuge to pellet the cells. Then, discard the supernatant. Add 100 microliters of diluted monoclonal antibody Ki-67 FIT-C to the pellet, then vortex and incubate for 30 minutes in the dark. After the incubation, wash the cells twice with 4 ml.